Hello, I am Karla Neugebauer. I'm the director of the Yale Center for RNA Science and Medicine, and I'm excited today to introduce Grace Chen, who's an assistant professor of immunobiology and genetics here at Yale. I want to start by acknowledging Frank Slack from Harvard, because he actually started our center here at Yale before moving on to Harvard. So it's always fun to share this slot. With our sort of sibling, I guess. So Grace, I just wanted to say a little bit about Grace's background. Sorry, I'm recovering from a cold. Grace did her BS in chemical biology at UC Berkeley and also did a PhD in chemical biology. So I needed to remind myself today that she has that strong background in chemical biology of RNA. And she discovered in her PhD with David Liu that NAD can be conjugated to RNA. And so that using mass spectrometry. So it's a really interesting exploration of the possibilities with RNA. And she, in fact, moved on to looking at circular RNAs when she postdoced with Howard Chang at Stanford University. So we were really excited to welcome her to Yale in 2019, bringing these unique perspectives on RNA. So I'm going to let her take it away and look forward to her talk.
Thank you, Carla, for that introduction. And I'm really grateful to the RNA Collaborative to be able to share our um, recent work. So here's that. And then this should be the full screen, I hope. Please, somebody let me know if they can't see the slides. So my group is interested in understanding how does a healthy cell maintain this vital balance between recognizing and detecting these foreign macromolecules, potentially pathogenic RNAs, to prevent infection, while also recognizing endogenous self molecules to prevent autoimmunity. And often these molecules look remarkably similar. And one class of these molecules that we study are circular RNAs. So you may recall that pre-mRNAs are spliced to remove the introns and bring together the exons. And many of these exons are joined together in RNAs that have a 5' cap and a 3' poly A tail. And I would say, 10 to 12 years ago, there were people who were studying a whole nother class of RNAs, and they found that there were RNAs that lack any end. And those ended up being these circular RNAs. And you can already see that there's a wide variety of different circular RNAs that can be made. Some that have only one exon, some that have, two or even more exons. And some of these circular RNAs can even contain introns or be solely intronic. These RNAs are now found to be highly dysregulated in diseases. So I just listed some of the different diseases here. That means that either the types of circular RNAs that are expressed change between, pathophysiology and healthy controls, or the levels of these different circular RNAs are changing between those two different states. So while I show that mammalian systems can express circular RNAs, they're actually found throughout the eukaryotic kingdom. And I've shown a phylogeny tree here, and I've labeled some of these different organisms. And you can see widely divergent species, essentially any organism that can undergo splicing has the capability to produce circular RNAs. I've labeled the metazoans in black here. And so these circular RNAs in humans can be at, thousands to tens of thousands of different ones, even within one specific cell. And they can undergo a variety of different functions. Um, some of them can regulate RNA or act as templates for proteins or act as scaffolds for proteins or sponge microRNAs as some examples of the types of functions that endogenous circular RNAs can have. More recently, circular RNAs have also been really used as a platform for therapeutics. And I've shown some of the sort of possibilities for how circular RNAs can be used as a new therapy or new treatment in blue. And my lab is really interested in understanding, how can we understand the basic science of circular RNAs and RNA biology to then integrate and develop new types of treatments to promote and improve human health. So in this process of thinking about, circular RNAs are expressed by eukaryotes, we wondered, are there non-self or foreign systems that also produce circular RNAs? And actually, if we had talked to, microbiologists, they would have told us that they knew about circular RNAs well before mammalian geneticists sort of, got interested in them. So the hepatitis delta virus contains a circular RNA genome, and this image shows one of the first demonstrations in nature that circular RNAs exist. More recently, people have found that DNA viruses, such as Epstein-Barr virus, Kaposi sarcoma-associated herpes virus, and the human papillomavirus, also encode circular RNAs. And so when we started the lab, we wondered, what about RNA viruses? So these are so viruses that have RNA as their genome. And because this was my first PhD student, we actually wanted to sort of stack the odds in her favor. And we picked the most sort of DNA-like virus that had an RNA genome. And you may guess that is HIV. So this figure shows a schematic of the different steps of the HIV life cycle. So the HIV is recognized by a receptor, then it enters, fuses with a membrane, comes in, and there's initiation of reverse transcription to turn the RNA genome into cDNA. The reverse transcription is completed within the nucleus, there's uncoding, and the HIV DNA is now integrated into the host DNA. In the nucleus, there is also transcription of both host and viral RNAs, there's splicing, and those RNAs then go out into the cytoplasm where there's translation into viral proteins, there's packaging, virion assembly, and then eventually release of the virion to then infect another cell. And we wondered, because transcription and splicing is so essential for the HIV life cycle, whether there might also be viral circRNAs that are produced in the nucleus during this time. So before I get into, the different experiments that we did to explore this question, I wanted to first acknowledge that there are some challenges associated with studying circular RNAs. And I think about this as trying to find a bunny, which has 2 ears, and a sea of cats, which has one tail. So first challenge is that circular RNAs are lowly abundant. By mass, they're only about 1 to 10% of mRNAs, which by mass is about 1 to 3% of total RNA. So we're already going from a small proportion of RNAs to an even smaller proportion of RNAs. Second, there's high sequence diversity between the circular RNAs. So there isn't a shared distinguishing feature that allows for their direct selection, such as mRNAs having a poly A tail that we can pull on directly. And third, they're mostly identical in sequence to their linear isoform. So if you recall how circular RNAs are made from pre-mRNAs, they're spliced from the same pre-mRNA as their linear RNAs, and mostly the linear isoforms are more abundant than the circular RNA. So I'll give the answer here. There's the bunny and the sea of cats. So in order to deal with these different challenges, we perform a series of negative selection to enrich for circular RNAs. So we'll take total RNA and perform a poly A selection, and that's actually our positive control, where we know that specific genes would be up or down regulated in response to viral infection. We then take the poly A minus fraction and then remove ribosomal RNAs. So the poly A minus, rRNA minus, fraction then undergoes RNA-SAR treatment. So either a mock treatment or RNA-SAR treatment for 120 minutes or 2 hours. So we call it RR0 and RR120. And so RNA-SAR is an exonuclease that degrades linear RNAs because it goes from the 5' end and chews all the way through to, chews from the 3' end all the way to the 5' end to degrade linear RNAs. because circular RNAs have no ends, there's no handle for it to sort of chomp onto. And so circular RNAs are enriched through this process. So here is the HIV genome that we were using. And our first control was uninfected cells. So we extracted the total RNA, enriched for circular RNAs from them, and then mapped it to the HIV genome. And we saw that there was actually, no sort of coverage by the tracks, suggesting that our uninfected control was truly uninfected. And that was good to see. When we looked at our circRNA enriched track sequencing results, we saw that it was across the whole genome. And that was really exciting to see that there might be sort of circular RNAs derived and encoded by HIV. And we also noticed that some of these dips relate to the known splice sites of the HIV. So again, giving us an indication that potentially there could be back splicing of the HIV genome to form circular RNAs. We then applied specialized bioinformatic pipelines to identify circular RNAs, which look for the backsplice junction in an inverted fashion. And we found, a bunch of different circular RNAs that were identified through this process. And we're looking for a circular RNA that is sort of lowly abundant in poly A plus samples because they don't have any ends and they shouldn't be enriched by poly A plus selection, and then upregulated in the RNase R enriched fraction. And this circ 5390 to 6044 sort of matched that those two criteria. So we started with that for further validation and study. So I'll be calling this circRNA circHIV for the rest of this talk. And it comes from the middle of the genome. And you might notice already that there's, part of VIF, some all of VPR, some TAT, and some REV encoded within this sequence. And we spent a long time trying to investigate whether or not there was translation from any part of the circular RNA. And thus far, we were not able to see sort of translation. So we treat the circRNA for the rest of this talk as a non-coding RNA. So the first step that we do with circRNA, once it's identified bioinformatically, is to perform experimental validation to make sure that this is a true bona fide circular transcript. One of the types of experiments that we use is divergent primers and then PCR. So divergent primers, if they're on a circular RNA, sort of produced from the cDNA produced from the circRNA, then it would be oriented towards each other and be able to make a productive PCR product. If the same divergent primers are binding to a cDNA produced from a linear RNA, then they would be oriented out and they would not produce a productive sort of PCR product. And we see that indeed for all of the poly A samples, either in the uninfected GERCAT or two models of HIV infection, they had sort of no signal. However, for the circRNA enriched fraction, we saw signal in the two HIV models, but not in the uninfected sample, sort of as expected. If we then run the PCR products on a gel, cut out that band, and perform Sanger sequencing, we can map exactly what our predicted backsplice junction with the Sanger sequencing results. And that was a really exciting day in the lab. So then we wanted to continue on to some other experiments. Here, we were checking whether or not the circHIV was RNase R resistant, and we would expect that it should be, and the linear RNA should not be, and that's sort of what we see here. So in the uninfected, we did not detect circHIV because there was no HIV infection. However, when we put in a plasmid that produces the entire circHIV, the HIV genome, we see indeed that there was enrichment with RNase R. In contrast, the linear HIV was significantly sort of downregulated upon treatment with RNase R, again showing the susceptibility of linear RNA to RNase R, this exonuclease, and the resistance of circRNA to RNase R. Next, we wondered, whether or not circHIV is actually expressed by full-length HIV virus. So previously, for our sequencing results and experiments, we had been using a model of circ-HIV. And here, this is sort of wild type circ-HIV with no marker for GFP on it. And we also used primary CD4 cells for this infection. And indeed, we were able to detect circ-HIV under these conditions, and that was also resistant to RNA-SAR, whereas the linear HIV mRNA was highly susceptible to RNA-SAR treatment. So far, all of these experiments I've shown were sort of, cell culture experiments, and we wanted to ask whether or not circ-HIV could be detected physiologically under natural infection. And so we were able to get the plasma from people living with HIV, and then we measured different sort of RNAs that would be present in those samples. So our negative control were 8 people who did not have HIV, and then we had we compare that with people living with HIV. So for 7SL, that was our negative control, we found that there was no enrichment in people living with HIV, sort of as expected. HIV-1 GAG was our positive control, and we would expect to see that people living with HIV would be expressing those RNAs, and indeed that was the case. The same thing was true for linear HIV mRNA, and we were really excited to see that for circ-HIV, there was also an upregulation of expression in people living with HIV compared to negative control, showing us that circ-HIV is truly found in natural infection. And so then we wanted to know where sort of cellularly was circ-HIV present. And we found that if we isolated the HIV virions, we were able to detect circ-HIV encapsulated within those virions. And so if we compare for, some of these different cellular RNAs, they are highly enriched in the cellular and not at all enriched in the virion. And that was also expected. And for 7SL, which we know is present in virion RNAs, that was our positive control, we saw an upregulation in the virion. And similarly, the case was true for circ-HIV. So now we know that circ-HIV is encapsulated within virions. And so at this point, now that we've established sort of the physiological relevance of circ-HIV, we wanted to come back into an in vitro, in cellular model to look at the functions of circ-HIV, if there was any. And here we had Jurkat cells that expressed shRNAs that targeted different parts of the RNA. So for circ-HIV, we had it target just the backsplice junction. And we used a GFP pseudotyped HIV virus, where GFP was then a readout for viral infection. And we found that if we targeted GFP, indeed, we had very low GFP expression. If we had a non-targeting shRNA control, we had relatively high GFP expression. And if we had circ-HIV that was targeting the backsplice junction, then we saw a decrease in GFP expression. And so this just quantifies both the percentage of HIV-1 expressing GFP positive cells we got knocked down with the shRNAs targeting circ-HIV backbone, as well as the GFP MFI also decreased when we had shRNAs targeting the circ-HIV backbone. So then we wanted to do the complementary experiment and ask where in the HIV life cycle was circ-HIV acting. And in this experiment, we transfected either the linear or the circRNA to model sort of what happens when the RNA is encapsulated into a virion and then infects the cell. We then have the HIV GFP infection, and then we collect different sort of macromolecules over the course of the HIV life cycle. So first we looked at viral entry, and we saw that there was no change in HIV genomic RNA, whether we had a sort of negative control, circ-M-cherry. This is not expressing or translating into circ-M-cherry. It's just a circular RNA with the M-cherry sequence on it to act like a non-coding RNA that is circular. Lin-HIV is the exact same sequence as circ-HIV, but it has a 5' hydroxyl and a 3' hydroxyl to model the sequence of the HIV. And then circ-HIV was our experimental test, and TAT is our positive control because we know where in the viral life cycle TAT should act. So here, this is the linear RNA 5' cap, 3' poly A tail, and we expect that it would be translated. So circ-HIV is not acting at viral entry. Circ-HIV is not acting at reverse transcription. Circ-HIV does not affect integration. And we were getting pretty nervous because we were doing these experiments sequentially as we were collecting the data, the RNAs. So the next step, we were excited to see that actually there was a change from our controls of Circ-HIV. And actually Circ-HIV was activating HIV transcription to similar levels as the TAT, which was our positive control. And we found that if we measure over time, that actually, TAT and Circ-HIV track really, really similarly to each other over this time course, whereas our negative controls were far below in terms of activating transcription. And so it was also, made sense to us that if we activate transcription, we would also increase viral protein expression because you had more transcripts that would undergo translation. And so at this point, we wanted to come back and ask, what is the mechanism for how Circ-HIV might be promoting transcription? And we did a really reductionist assay where we had the HIV promoter just upstream of a firefly luciferase, and we either put in linear RNA or circRNA just by itself. And we found that independently of TAT, Circ-HIV was able to activate transcription above its negative controls. It is lower than TAT here. And so we wondered, given they were both acting at the same stage of the viral life cycle, whether or not they might actually act together in synergy. And the answer is yes. So if we have TAT alone or Circ-HIV alone, it was less than if we had Circ-HIV plus TAT. And again, our negative controls of Circ-M-Cherry and Lin-HIV had no sort of activating abilities here. So I'll remind you that TAT actually stands for the trans activator of transcription, and it's known to be the master regulator of HIV transcription. And so we wondered whether or not TAT might directly interact with Circ-HIV. And so here we used an antibody to enrich for the TAT complex and any bound RNAs, and then we performed RT-qPCR. And we found that our positive controls behaved sort of as expected. Our negative controls behaved also as expected. And then we saw that Circ-HIV indeed does interact directly with TAT inside the cell. So then we wanted to do an in vitro binding assay to see if we can determine what portion of Circ-HIV would interact with TAT. So here's the full length, and we perform in vitro transcription where we add on biotin into our RNAs. We can then make them circular. We use streptavidin beads to immobilize those RNAs, and then we incubated them with the protein TAT. So we found that TAR was our positive control, and indeed, it still binds TAT. Circ-M-Cherry and Lin-HIV, as in all of our previous assays, had no ability to bind TAT, and Circ-HIV, we were able to see indeed bound TAT. So then we looked at whether or not Circ-HIV and TAR, which are our positive control for interacting with TAT, actually competed for the same binding site on TAT. And so we either increased the amount of TAR and kept the level of Circ-HIV present and stable, or increased Circ-HIV and kept the level of TAR stable. And we found that as we increased one RNA or the other, there was no impact on the other RNA for binding TAT, suggesting that they do not compete for TAT binding. We then mutated different windows along the Circ-HIV to see if we could sort of scramble and identify a region that would enable or disable Circ-HIV to interact with TAT. And indeed, we found that if we mess with mutant 2, which is this sort of green window in the second region, we abrogate TAT binding. And so this suggests there's something about that region that either its own structure or its structure scaffolding for the rest of the Circ-HIV enables TAT binding. And so here is our current model where we think that Circ-HIV either has independent activity to promote and drive HIV transcription, and actually in combination with TAT, synergizes with TAT to really activate and enhance HIV transcription. So there's still a lot of open questions about sort of the Circ HIV mechanisms and its interacting components and its partners, and where in the RNAs it might be sort of regulating which part of the life cycle. And I just want to spend the last couple minutes just to mention about technology development. So in the Circ HIV field or Circ RNA field, often it feels like this biological question depends on us building the track for technologies. And I just want to draw your attention to one step I mentioned earlier, which is rRNA depletion. So if we calculate how much RNA we actually need to deplete ribosomal RNAs from, and then calculate how much all of that would cost, it is exorbitant and would be really hard for us to do on, say, a modular R01 budget to continue some of this work. And so that's why in the lab, we developed a method called rRNA removal by RNase H, where we design probes that bind to ribosomal RNAs to deplete that for really high efficiency and high concordance with commercial kits and for a fraction of that cost. So we're happy to share aliquots of this probes if you're interested in testing it for your application. And then I also want to comment, I talked about producing circular RNAs for testing and modeling some of these biological questions. And we also have some, new ways to produce really precisely designed and generated circular RNAs. So I'm really grateful for the people who have done this work. Priska and Lee Chong really worked on the Circ HIV story. Amrita has driven many of the technologies that we've used in the lab. And I want to acknowledge the other people in the lab who have worked on projects that I didn't have time to get to, and our funding sources. So thank you so much for your attention. I'd be happy to take any questions.
Thank you, Grace. That was great. Very interesting. I love how you focused on that one story and took us through it. I actually have a question about those wonderful experiment where you showed the synergy between the circle and the cat. Or, yes.
Yep.
And my question was, did you ever try a like order of addition experiment where adding one first and then the other had the synergy, whereas if you added, do you see what I'm saying?
Yeah, absolutely. There are a lot to do in this sort of experiment here. So this is
Yeah,
just with, all of the RNAs by itself. Here's the RNAs with TAT. And so
right.
we have done a dose titration curve, and we see sort
Okay.
of, a similar ramping in responsive activity. And we want to do some of these sort of order of addition. How long do
Right.
you wait between adding the next one to see sort of the impact here? Absolutely.
Okay. And then there's a question in the Q&A that maybe just as an issue of clarification. Shalini is wondering if biotinylation promotes circRNA formation. Um,
Yeah, so I guess that's about the sort of just efficiency of
yeah.
making the RNA.
Mhm.
We run this in parallel with unmodified, and thus far we haven't seen a huge difference between circularization efficiency. We add biotin to, less than 10%. So it's not that
Mhm.
every single nucleoside is modified. We haven't seen a huge difference in our hands.
Is that the U that you used? Or
I think
what
that's right.
nucleus?
Biotin U, right.
Okay, interesting. Because biotin also, inhibits transcription. So yeah, I guess it's just one of those things you have to play around with, make sure that you get
Yeah, so
as
here,
much.
when we add in biotin, this is a totally
Mhm.
biochemical assay. So it doesn't go into a cell and there's no transcription happening here. It's just looking at
Right, right.
direct
You're
binding.
just trying to
Absolutely.
make your reagent. Yeah, yeah,
That's right.
about.
In a way that we can
So
detect.
is there so one question is like how general this is. I don't know if you touched on that at the end and I missed it. There's all these circRNAs that are reported and they in principle could have similar or completely different functions. What's your feeling?
Yeah, so I think there are probably group effects and also individual effects. So data that I didn't show is that if we take the HIV virion and just assay how many circular RNAs are in there, there's so many different types of circular RNAs in there. There are also host circular RNAs in there. So
Mm,
this could be sort of a group type effect where having some cluster of circular RNAs has some positive effect on HIV
I
viral
see.
infection.
Okay, got you.
This circ HIV sort of, promoting HIV transcription, I think is not a global
Mm,
generalizable function of all circular RNAs.
got you. Interesting. Okay, can't wait to hear the next chapter. So we should wrap up because it's 4:30 and turn it over to the next speaker. So thanks again, Grace.
Thank you.
Fantastic. Thank you very much, Grace and Karla. So great to have you all join us at the RNA Collaborative today. My name is Frank Slack, and I'm the director of the HIRM. Today, our speaker is Dr. Elena Piskounova. Dr. Piskounova is an assistant professor at BIDMC's Cancer Center and Harvard Medical School. She received her combined bachelor's and master's degree in molecular and cellular biochemistry from the University of Oxford in the UK, and went on to complete her PhD training in biological and biomedical sciences in the laboratory of Dr. Richard Gregory at Harvard Medical School. She then did postdoctoral work with Dr. Sean Morrison at UT Southwestern Medical Center, where she discovered oxidative stress as a major barrier of metastasis and identified key metabolic adaptations required for survival of metastasizing cancer cells. She then started her independent laboratory at the Maya Cancer Center at Weill Cornell Medical School before moving to the BIDMC Cancer Center a few years ago. Her research aims to identify the molecular mechanisms of translation that enable survival of metastasizing cancer cells that can be targeted therapeutically in metastatic disease. And in particular, her recent focus is on understanding the role of tRNA modifications in regulating codon bias translation and identifying novel structural RNA elements that modulate translation under stress in metastatic cells. So welcome, Dr. Piskounova. The stage is yours.
So thank you, everybody. Now that you can hear me. So I'm going to tell you some of the research we've done on tRNA-driven translational reprogramming and metastasis. So my lab is interested in understanding metastasis because, of course, it's a devastating consequence of cancer for many patients, but it's also a highly inefficient process. Very few cells can detach from the primary tumor, enter the bloodstream or the lymphatics, and become metastatic tumors in vital organs. So we aim to understand why are some cells successful. And as Frank mentioned, as a postdoc, I found that oxidative stress limits distant metastasis of melanoma, and only successful metastasizers that increase their oxidative stress resistance are able to actually colonize distant sites. And this since has been shown to be true in melanoma and other cancers, so it seems to be a more general phenomenon of the metastatic cascade. So since then, we have found that micrometastatic lesions have much higher levels of oxidative stress in PDX models of melanoma, both cytoplasmic and mitochondrial oxidative stress, compared to macrometastatic lesions. Interestingly, we see that proteotoxic stress markers show the same pattern where micrometastatic lesions seem to have higher levels of these stress markers compared to macrometastatic lesions. So it seems that micrometastases exist in this much more vulnerable state. They're much more stressed, and we think they're
I think we might have just lost your audio again.
Again? No.
Okay, now, yeah, it's back. It's back now.
Okay, sorry. I think a more general, something that's more generally accepted in the field is that metastasis is limited by many stressors, starting with anoikis, hypoxia, interactions with the immune system, and of course, any therapy that is being administered to the patients. So metastatic cells have to really survive under stress in order to become successful colonizers. And in order to successfully metastasize, cells have to adapt through different mechanisms, including epigenetic, transcriptional, metabolic pathways. But we were looking for the mechanism that would rule them all, and we settled on translational reprogramming. And translational reprogramming is also a way for a cell to rapidly respond to environmental cues. So I'm going to tell you two short stories, one on regulation of stress granule formation by tRNA wobble modifications, and one on activation of GCN2 by mitochondrial tRNAs in the cytoplasm. So when we started thinking about translational reprogramming, we started reading the literature and found that in model organisms such as yeast, there's a phenomenon that is very well established, where cells in optimal conditions have a steady state tRNA pool, they perform translation and produce proteins. However, cells under stress modify specific tRNAs in a very particular way to induce codon bias translation and change the proteins that they're making. So we hypothesized that tRNA anti-codon loop modifications, together with codon bias translation, increase stress resistance in metastasizing cells. So the way this looks at the molecular level is that we're looking at the anti-codon loop at position 34, or the wobble position, which base pairs with the third position of the codon. And when there's a uridine in the wobble position, it is quite promiscuous. It can base pair pretty much with anything. But uridines are actually highly modified by this cascade of enzymes, starting with the elongated complex, then methylated further by LQBH8, and then thiolated by CTU1 and 2. And what this does is that this modification, MCM5S2U, limits the base pairing to A-ending codons and increases the translation of A-ending codons. And this can have a widespread effect on the proteome because at least 4 different codons are affected. They exist in these split codon boxes, and it can affect lots of different proteins. So when we started the work, we wanted to really see if this phenomenon exists in our models. So we looked at the TCGA data in melanoma patients, and we looked at the codon preference in transcripts enriched in metastases compared to primary tumors. You can appreciate that all of the A-ending codons are enriched in the metastatic transcripts. And the converse is true, that G-ending codons are enriched in the transcripts in the primary tumor, not in the metastases. So we think that there's codon bias translation happening in metastatic melanoma. So to get at the mechanism, we knock down the elongated complex, one of the components of the elongated complex. And what this does is actually destabilizes the whole complex. It's a complex of 6 proteins. And we see the decrease as expected of the modification. And we also see an interesting increase in the S2U modification. We're not sure what this means, but we're still following up on that. But we tested in vitro phenotypes such as migration and invasion, and we see that cells migrate and invade less when we lose the elongated complex. But of course, for us, it matters what happens in vivo. So we knock down the elongated complex again in a PDX model of melanoma, and the primary tumors were completely unaffected. They grow perfectly normally. But we see a significant decrease in metastasis in the organs of these mice. We can phenocopy this by knocking down LQBH8, which is the second step of the pathway. Again, primary tumors couldn't care less, but we see loss of metastasis in the organs. So we think that this particular pathway, this tRNA modifying pathway, is particularly important and specifically important in metastasizing cells, not highly proliferative cells in the primary tumor. So to get at the mechanism, what we did was we created these reporters, which are GFP reporters, with either G-ending codon bias or A-ending codon bias. And if we knock down the elongated complex, we can show that only the A-ending GFP is reduced compared to the G-ending GFP. So we know that the A-ending translation is impacted. We next did this, I think, one of the cooler experiments that we've ever done, where we correlated the fluorescence of these codon biased GFPs with the migration of the cells in vitro. And you can see that A-ending GFP fluorescence correlates nicely with migration of cells, whereas G-ending does not, or the just regular luciferase GFP. So this means that A-ending translation correlates with this metastatic phenotype of migration of melanoma cells. We performed ribosomal profiling, and we see stalling of the ribosomes at the A-ending codons, as we would predict. Interestingly, we found that leucine TTA also shows stalling, so we think there might be other A-ending codons which are impacted by this pathway. And then, because we cannot do ribosomal profiling in vivo, what we did was proteomics, comparing primary tumors and metastases from the PDX model, then looking at the amino acid composition of these proteins, and then calculating the codon bias of these proteins, kind of going backwards to the RNA. When we looked at the proteins, which are shown in yellow, these are the codon biased proteins, we found that components of stress granules, such as G3BP1, G3BP2, and PABPC1, were highly enriched. And this was true for 2 different models of PDX melanoma. And this was only happening at the protein level, because at the mRNA level, these components are not changing. And we also performed GO-term analysis, which I'm not showing you. So stress granules is a way for cells to pause translation. At a steady state, translation initiation complex, which includes eIF2 alpha, is assembled on the mRNA, However, if there's a stress stimulus, eIF2 alpha gets phosphorylated in response to many different stresses. And what this does is it leads to disassembly of the initiation complexes. But instead of degrading all the machinery, what cells do is they assemble them into these non-membrane bound organelles called stress granules, which are nucleated by proteins such as G3BP1 and 2, and it kind of keeps everything paused. So that when the stress stimulus is resolved, the cells can either just disassemble the stress granules or they can degrade them through autophagy. And this allows them to recover their protein translation. So we also looked at codon bias of these components that we found, G3BP1, G3BP2, and PABPC1, and they're all codon biased for the A ending codons. You can see they're all above average for the codon bias. So we think that they themselves are regulated by this pathway. So next, what we did was we knocked down the elongated complex and saw a decrease in G3BP1, G3BP2, and PABPC1. You see this more pronounced when we induce stress granules with sodium arsenite. We can also overexpress the wild-type coding sequence of G3BP2. We've consequently done this with G3BP1 as well. And again, when we knock down the elongated complex, you see a reduction of the overexpressed G3BP2, and this is not changing at the mRNA level. But if we overexpress a G codon bias coding sequence, you can see it's expressed much more robustly and it's not responding to the elongator loss. So we think that this is, again, a codon bias phenomenon. At the cellular level, we can induce stress granules with sodium arsenite, and then when we knock down the elongated complex, we observe fewer stress granules assembling in the cells. We now knocked out and down G3BP1 and G3BP2, and we see a decrease in migration of the cells and a decrease in invasion of the cells when they lose these proteins. But importantly, in vivo, we also see pretty much complete loss of metastasis if you lose stress granule components. The caveat here is that the primary tumor growth is slowed down and also reduced a little bit, but it's not as dramatic as the metastatic phenotype. So to summarize this part, what I've shown you is that this tRNA modification pathway that controls the wobble uridine modification seems to regulate stress-related codon bias proteome. We focused on the stress granule formation components from this proteome, and we think that this pathway is very specifically involved in metastatic colonization rather than primary tumor proliferation. So now I'd like to change gears a little bit and tell you a very new story. It's very much an emerging story from the lab on the activation of GCN2 by mitochondrial tRNAs in the cytoplasm. So we have been considering the integrated stress response as a mechanism of translational reprogramming for quite a while because it's really key to reprogramming translation under stress through phosphorylation of eIF2 alpha, which I have already mentioned. So eIF2 alpha can get phosphorylated by 4 different kinases in response to different stimuli. GCN2 senses deacylated tRNAs, PKR senses double-stranded RNAs. HRI traditionally has been thought of as responding to low heme, but more recently has been discovered to respond to mitochondrial dysfunction, and PERK senses proteotoxic stress. So when eIF2 alpha is phosphorylated, cap-dependent translation is blocked, but stress response translation continues, or actually is induced, and one of the central components of this is ATF4, which can then go on and induce a wide transcriptomic response for stress response genes. So when we looked at the ISR in our system, in the PTX system of melanoma, we see that components such as ATF4 and phospho-F2 alpha are highly upregulated in micrometastatic nodules. They're also upregulated in macrometastatic nodules compared to the primary tumor. We see transcriptional changes in metastases as well, but importantly, ATF4 is not changing at the mRNA level. This is a purely translational response. We used a reporter of ATF4 where the N-terminus of ATF4 is fused to an HA tag and scarlet so that the translation of ATF4 can be read out as scarlet fluorescence. We put this reporter in vivo and we see that there's more ATF4 ATF4 translation in metastases and circulating tumor cells, which was really exciting to us. But of course, we always come back to the question, is this a functional phenomenon? Does it play a role in metastasis in vivo? So we can inhibit ISR with ISRIB, which blocks phosphorylation of eIF2 alpha. And at different doses, ISRIB does not affect primary tumor growth. The tumors really grow perfectly normally. But we see a reduction of metastasis at different doses of ISRIB, and we see a reduction in circulating tumor cell frequency as well. So this really means that ISR is playing a functional role in allowing cells to colonize vital organs. So then the question became, well, what is the upstream signal that activates ISR? And as a postdoc, I looked at mitochondrial dysfunction in metastatic nodules, and I saw that mitochondrial mass was changing, mitochondrial potential was changing, and metastatic nodules had very high levels of mitochondrial ROS. We also looked at the structure of the mitochondria, and you can appreciate that the primary tumor mitochondria are much better looking than the metastatic mitochondria. The metastatic mitochondria are round, they're bigger, they're kind of bloated, they're more damaged. So we think mitochondrial damage may be influencing ISR activation during metastasis. And as I mentioned before, HRI in the last 5 years has been discovered to respond to mitochondrial dysfunction through this pathway, going from OMA1 to cleaving DELI1, and then DELI1 binding HRI and activating ISR. And this pathway has been implicated in breast cancer metastasis to the lung. But in our case, when we looked in our model, we saw that phospho-GCN2 was the one that was actually changing the most in metastatic nodules. We're not sure about phospho-HRI because there is no antibody for phospho-HRI. We think this upper band might be phospho-HRI. We're still figuring it out. But other kinases such as phospho-PERK and phospho-PKR were not changing. So we were curious about the fact that there's phospho-GCN2 activation. And we induced mitochondrial dysfunction using oligomycin A to induce ISR. As you can see, ATF4 and CHOP all go up. When we knock down GCN2 in these cells, we see a reduction of ISR induction in response to oligomycin A. When we knock out HRI, we also see a reduction of ISR, but it's only when you combine them together do you lose ISR induction entirely. So we think that maybe these two kinases are converging and activating ISR in response to mitochondrial dysfunction. We can induce mitochondrial dysfunction with oligomycin A, but we can actually phenocopy it by using pyacinin, which is a pro-oxidant. It's a global pro-oxidant that causes massive amounts of oxidative stress. And we see that mitochondria become depolarized in response to both. So we use pyacinin to model oxidative stress in vitro. So when we knock down GCN2 and induce ISR with pyacinin, we see less ISR being induced, and we also see less ISR being induced in response to oligomycin A as well here. We think the kinetics differ a little bit because the timing of the treatment is different for these different compounds. But of course, what is the molecule that activates GCN2? And this phenomenon has been pretty well described before, that in response to mitochondrial dysfunction, mitochondria can leak components into the cytosol. And this can be mitochondrial DNA or mitochondrial RNA. So we speculated that maybe mitochondrial tRNAs also get released into the cytosol, and maybe they can activate GCN2. We performed sort of a general experiment where we inhibited mitochondrial transcription with IMT1 and showed that levels of mitochondrial tRNAs go down and mitochondrial RNAs also go down. But we also showed that there's less induction of ISR with IMT1 in response to oxidative stress. So there's a link between mitochondrial transcription and ISR that we can establish. We performed fractionation of cells either in control conditions or under oxidative stress induced with pyacinin, and we actually detected some mitochondrial tRNAs in the cytosol, in the cytosolic fraction. Not all tRNAs, but some tRNAs are definitely enriched. So we think mitochondrial stress or oxidative stress induces release of mitochondrial tRNAs into the cytosol. We did the same experiment in tissues where we did fractionation in metastases and primary tumors, and we can see more mitochondrial tRNAs in the cytosol in metastatic tumors than the primary tumors. So we think this is actually happening in vivo. So we wanted to rescue this effect of release of mitochondrial tRNAs in response to oxidative stress with antioxidants. So we used N-acetylcysteine. And what was interesting was one particular tRNA, this mitochondrial tRNA lysine TTT, was rescued with antioxidant treatment. So that was really peculiar to us because there seemed to be some specificity that is happening. And I think the key experiment that we have done now is a GCN2 clip to really show that there's physical interaction between GCN2 and mitochondrial tRNAs. And what was really exciting to us is that this mitochondrial tRNA lysine TTT was the one tRNA that was associated with GCN2 in response to oxidative stress. So it seems like that could be our candidate that activates GCN2 under stress conditions. In terms of cellular phenotypes, we can knock down GCN2 and show that we can block migration of melanoma cells in vitro. It's a very dramatic phenotype. And we have since done some pilot experiments where we have knocked down both GCN2 and HRI in vivo. Again, I'm showing you the same thing. Primary tumors really don't care if we knock down these proteins, but we see a reduction of metastasis in both cases. So we think this is truly a convergent signal from both kinases that is happening in vivo in the metastatic cascade. So this is very early on, the work is ongoing, but this is our hypothesis. We think that oxidative stress that metastatic cells experience during the cascade as they migrate and invade leads to mitochondrial rupture, which leads to release of components of mitochondria, including tRNAs, into the cytosol. These mitochondrial tRNAs, maybe very specific tRNAs, bind and activate GCN2, which induces the integrated stress response, which promotes the survival of metastasizing cells and colonization of vital organs. So with that, I'd like to acknowledge Riley Hughes, who did all the work on the stress granules, and Sujin Kim, who's now doing the GCN2 work and mitochondrial tRNAs, and I will be very happy to take questions.
Great. Thank you very much, Elena. That was fascinating. Just a reminder to our audience, please type your questions into the Q&A, but I will get started whilst people are thinking and typing. It was fascinating. I mean, two sort of independent stories, but I wonder, could there be a connection between the two? Do you know whether this mitochondrial stress or these tRNA releases can sort of activate stress granule, or where do these tRNAs go? Do they go into granules of any kind? Do you know?
I'm not actually, that's a great question. I'm not aware of anyone having looked at tRNAs being sequestered in the granules, but definitely the link that maybe I made too quickly is that phosphorylation of EIF2 alpha induces the integrated stress response and formation of the stress granules. So those two are very much linked processes, and they would basically interact at that level. So they would be happening concurrently, I would assume. Yeah.
All right, cool. And do you have a sense of whether this tRNA, I guess I can say leakage into the cytoplasm, occurs in any sort of physiological context other than metastasis? For example, is there a time in development or a time during, organismal physiology where something from the mitochondria might signal to the cytoplasm?
Yeah, that's a great question. I think the one thing that metastasis field always goes back to in terms of a parallel process that happens physiologically is wound healing, because cells have to migrate and invade during wound healing, and that sort of can stress them out. So it could be that could be a context in a more non-cancer related context. And I think also there's a fair amount of literature on mitochondrial stress in neurodegenerative diseases and other non-cancer contexts where I think that could play a role.
I was wondering whether, because you found this in melanoma, that maybe there was something around neural crest cell migration or something where
exactly, yes.
they're moving quite extensively in the early developmental stages.
Yeah, I actually think that's probably exactly the context, where it's coming from.
Yeah, very cool. And any sense of whether this is sort of a passive leakage, or do you think that there's some sort of transport mechanism that moves tRNAs out of the mitochondria?
Yeah, we sort of started with the idea that it was passive, that it was just damaged mitochondria and basically mitochondrial damage being relayed to the cytosol that, hey, there's damage happening, so we need to do something about it, because that's where the literature on release of mitochondrial DNA and mitochondrial RNA has been looking. But when we saw that specific tRNAs are being released, that could suggest that there's an actual controlled process that is happening, and it's not just a general dumping of the components into the cytosol.
Yeah, very cool. Karla, you can just unmute and ask your question. Go ahead.
I feel so privileged. Yeah, I was wondering if somehow thinking about the evolution of this and also other organisms that might use this. Would this play out, would the codon usage issue also play out in the context of temperature changes?
Absolutely, yes. So in yeast and in other organisms where people have done heat shock experiments and
Mhm.
other stresses, osmotic stress,
Yeah.
so where there are other stresses that you can induce, there's definitely a role for this, specifically for this uridine modification pathway. Yeah, absolutely.
But that's a stress response, but I was sort of wondering more about tuning, like if the temperature itself could have an effect on structure base and base pairing that could be significant enough that you would kind of emulate that, over evolution by using modifications as a different way of tuning strength.
Yeah, that's a
Do you know what
really interesting idea. Yeah. So more the thermodynamics of it. Yes.
Yeah. I just, one of the things I wrestle with is whether those sort of temperature changes are like relevant, but ectotherms, they go plus and minus 30 degrees kind of, so or almost 20 degrees. But like, that's a huge temperature range to have to translate a protein accurately. So you do
Yeah,
wonder
that's
if they start, using different amino acids. Um, yeah.
Elena, we have a question in the Q&A. Do you have a sense why it's only empty lyse TTT? That's the one that binds and activates GCN2?
So we have a little bit of a sense. We've done a little bit of work with AlphaFold, modeling the binding of the mitochondrial and cytosolic tRNAs to GCN2 or part of GCN2. GCN2 is quite huge. And it seems that the mitochondrial lysine TTT forms a lot of favorable bonds, just maybe the structure is favorable for binding to GCN2. We compared it to mitochondrial tRNA for phenylalanine, which was not changing. And the binding modeling said that it was less likely to bind than the lysine tRNA. So I think it's something to do with the structure. Mitochondrial tRNAs are different structurally from cytosolic tRNAs. They also modified a little bit differently. So maybe it's something to do with how the tRNA folds and is able to be a substrate for GCN2.
I was going to follow up with that and ask whether there's any evidence that mitochondrial tRNAs get charged, and can they actually engage in translation in the cytosol?
I don't think I'm not aware of anyone showing that. And I think because they are structurally different and modified differently, I think they wouldn't fit into a cytosolic ribosome. At least that's my sense. But I'm not sure that whether anyone's ever done any work on that.
So I was going to say maybe the GCN2 is just blocking them from engaging in some spurious function that they shouldn't be doing. All right. Well, I think there are no more questions in the chat and the Q&A either. So I think we'll end by thanking all of our speakers today. Thank you very much. I believe there is a brief announcement about the next RNA Collaborative meeting in the chat, which I think says that it's going to be July 8th at 10 o'clock in the morning Eastern time. So it'll be 4 o'clock European time. It'll be hosted by the Institute for Molecular Biology in Mainz and the RNA Research Center in Poznan and Poland. So please, everybody, join us again next month. So thank you very much, everybody. It was great seeing you all. Bye-bye.